both adult and pediatric populations. Before joining Pfizer, Meg served as the research advisor in global statistical sciences at Eli Lilly and Company and as a mathematical statistician at the FDA. As a fellow of the American Statistical Association, she has led several industry initiatives, including scientific work groups on statistics in pediatric drug development and the statistical perspective on AI ML in pharmaceutical innovation task force at the Biotechnology Innovation Organization and contributed to the European Forum for Good Clinical Practice Children's Medicine Working Party. It provided guidance on the inclusion of adolescents in adult research. She has held various positions in the biopharmaceutical section of the American Statistical Association and the Society for Clinical Trials. In addition to her leadership roles, Meg serves as the editor-in-chief of the Journal of Biopharmaceutical Statistics. She has authored over 70 peer-reviewed scientific publications in the areas of Bayesian methods, evidence synthesis, wind statistics, causal inference, and pediatrics, among others. I'm also proud to say that Meg is a batchmate of mine. She was our BS Mathematics Program awardee way back a few years back. Without further ado, to share with us about the role of quantitative statistics scientists in pharmaceuticals industry, this is Dr. Margaret Gamalo.
Thank you very much, Ray. Yes, again, I'm very happy to be here again and talking about the role of quantitative scientists in the pharmaceutical industry. I made this a little bit more general because while I am a statistician, the role of statistics in the pharmaceutical industry is also changing. The fact that there is widespread use of AI as well as computer algorithms in many of the things that we do. And this is why I thought that it's not necessarily just statistics as a discipline, but more like a role on of being a quantitative scientist in the development of medicines. Can you go to the next slide, please? So statisticians, and again, as well as anybody in the quantitative field, are essential across the entire drug development value chain. And that starts all the way from discovery, lead identification, as well as preclinical studies, all the way to first in humans, phase 2 trials, which are proof of concept, pivotal trials, which are phase 3 trials, and then the post-marketing requirements, surveillance, and health technology assessments. As you can see in here, I have dotted each of these phases of development with where quantitative scientists can actually be involved. Statisticians in particular are involved in almost everything. We do a lot. Disease modelers, non-clinical statistics are usually involved in the discovery as well as lead identification, preclinical stage, biomarker development. Drug safety, on the other hand, are involved in almost everything too, starting from preclinical all the way to post-marketing. And then we have some benefit risk assessments, epidemiology, market access, health economics, real world data. We also have psychometrics. Psychometrics are used in the patient-reported outcome development, as well as a clinical reported outcome development and validation, making sure that these measures are actually measuring what a patient feels, functions, and survives. Quantitative scientists are involved in portfolio management as well, particularly in understanding what would be the value of the product and how we can make sure that the development that we have has a return of investment in the future. So that's where portfolio management comes from. And of course, data scientists, which uses integrative methods for understanding what our potential insights into multiple data sets are here too. Next slide, please. So this is where a statistician or any quantitative scientist, again, I'm trying to blur statistics as well as quantitative scientists because it is possible that quantitative scientists can perform as a statistician as long as the required skill is there. There is really this blurring of disciplines, but maybe not as a role. So the role is with respect to for the development, but then the discipline and the training could be different as well. Meaning that the training could be a little bit unconventional to a statistician, but then in the end can function as a statistician in the cross-functional medicines development. In here, I broke down several parts of a clinical trial, starting from planning, startup, execution, reporting, submission, as well as pricing and reimbursement, and potentially as well pharmacovigilance. In general, we are already involved in so many things, including, for example, SDSA, which is programming, from the very get-go of trial development. So from clinical development plans, so this is how we are going to create studies that in support of a submission to the FDA. So that's the clinical development plan. The asset development plan, meaning what are the potential indications for which we will develop the assets with? For, it could be, let's say, atopic dermatitis, but then this is also being used in, let's say, allergic rhinitis, then that is involved in the asset development plan. And the more granular one is the protocol development. And in general, in many of these things, we start with due diligence and data mining because we wanted to understand several things that would be helpful in the clinical development as well as the writing of the protocol. One would be endpoint development, one would be disease progression, one would be inclusion, exclusion, and one would be how the monitoring of these patients happen during the course of the trial. At the same time in here, we also have to start working on the program level standards analysis and reporting and modeling as well. So both a statistician or a quantitative data scientist in programming will have to start doing this as well during startup. Endpoint assessment training is also one because, for example, if a patient does not know how to measure the disease in a PRO, then most likely it will just be noisy at the end of the day when we analyze these things. So patients and clinicians have to be trained on the endpoints in such a way that we get something like no garbage in, garbage out. We also have data review plans, which is in general the way that we would be monitoring the data as it comes in the trial. Right now, there is a lot of, there's always fraud involved in clinical trials just because patients are being paid, sites are being paid. And so therefore, there's always going to be conflict of interest where a particular site or a particular patient is remunerated and therefore would have the propensity to commit fraud just because money is involved. And so for us in the pharmaceutical industry and for sponsors, we have to make sure that we are monitoring our data very well. And again, in such a way that this will, that the data is in line with good clinical practices. We are protecting the safety of the patients. At the same time, we are submitting data that is trustworthy to the regulators in support of a particular indication. So what I'm going to be talking about is just this area in red currently. So I'll talk more about it in the next few minutes. And then towards the end, I'm going to give some examples of some specific things that we are actually doing. So next slide, please. So I've divided, okay. I divided certain things that we are doing in every single activity, right? For example, data mining. In data mining, a statistician or a quantitative scientist performing as a statistician would start working with a target product profile for a product and a meta-analysis of competitor products for which we would benchmark a successful program with respect to this meta-analysis. So what does a target product profile mean? So when we develop a particular product, we wanted the product to have certain characteristics, and we wanted the product to have an efficacy that is usually at par or even better than the existing product. And there are different ways for which a product will differentiate from what is existing from the market. One would be efficacy, one would be safety, one would be ease of administration, for example, an injection as opposed to an oral drug, or it could be that there is a particular biomarker that would help increase the activity of the drug in that particular patient, and therefore efficacy if the patient is positive for that particular biomarker. So one of the things that we have to do is, create this target product profile and perform a landscape analysis and the effects of all the drugs out there for a particular indication, and then benchmark what would be a good product to develop. If the product at the end of the day, after a particular trial, will not be as good as what is this benchmark, then potentially we will no longer develop the product, because it's, as we will be seeing, it will not be competitive in the market. So we always have to be better. No, it's not like we're just going to be creating me too products every single time. During analysis and reporting and modeling and simulation, we also have to do a lot of due diligence because we wanted to understand what is the placebo rate? How does placebo behave in these particular trials? And how do we know that we are going to be better than it in multiple of these endpoints? Usually what we do is that we gather data from multiple sources. One would be Transcelerate, which is a repository of placebo data, and one could also be real-world data, which is coming from electronic health records. Electronic health records, of course, is a treasure trove of data, although it's a little bit messy, but nevertheless would be able to provide us with certain things regarding how placebo actually progresses over time. So, for example, a patient would be given a standard of care, which is what we would think to be placebo, just because most of the time when we are developing medicines, it's on top of what is already standard of care, right? And then how does this standard of care, if you look at the electronic health records, how do patients move? How do patients behave? Are some of the patients actually improving, and so on? We can use this, the real world data as well in relation to Transcelerate, which is placebo, to create digital twins. So there are actually ways of creating twins that look like, potentially how an active drug would actually behave. So these are just some of the things that we are doing on this end. Next, please. Is it moving?
Possibly. Oops, sorry. Oh, si Chair. Ayan, share na. Naka-share na. No, not yet. Sorry.
No worries.
Jerry? No, not yet.
Actually, but not full screen. Picture. Full screen. Oh, but it's not. I just need to.
All right. We are now in here, no? Okay. The other one that is very important in this particular stage is on the operating characteristics of the design. We wanted to understand, for example, what would be go, no go probabilities. So if we have this particular compound with this chemical structure, with this absorption, distribution, metabolism, and excretion and toxicology. And if we are seeing early phase trials that seem to be, promising, how does that translate? How can we predict subsequently what will happen in phase 2 and what will happen into phase 3? And the reason that's very important, it's because we have several compounds that are competing. And therefore, if we are able to understand which of the compounds will likely be successful in the market in the end, then we can restructure our resources in such a way that those compounds that are of high priority will be given the most resources. So some of these things are actually happening as well. Inclusion-exclusion criteria. With the age of AI, this has become very different. Before, inclusion-exclusion is the realm of a physician to be able to determine. But because of artificial intelligence, many of these things can actually be quantitatively optimized using Shapley values as well as line, which is another measurement of importance of inclusion-exclusion. So all of a sudden, a statistician can actually behave or can advise a physician on what would be a good inclusion-exclusion that will isolate the effect of the drug instead of muddy it when the inclusion-exclusion, when we are including patients that potentially have a lot of confounders and effect mediators. Another one on the right, which that we are also working on as a statistician or a quantitative scientist working as a statistician, is on recruitment and exposure accumulation. Why is this very important? The value of a product, the value of a drug, is usually dependent on how fast you can put it in the market. And the reason for that is there is a renegotiation period for which after that renegotiation period, the price of the drug goes down. There is also a period for which the loss of exclusivity happens. By that, in that particular case, a generic product can come in. So therefore, we really wanted to understand whether we can recruit a particular trial as soon as possible, rather than if it is, taking a very long time to recruit, by which that would eat up on the value of the product because there would be less time to promote it and to market it. So recruitment is one of the things that is very important in the role of a statistician, being able to determine when the trial stops, when is adequate safety exposure accumulated, and when the submission is going to happen, and when is approval potentially going to be achieved with a regulatory agency. And we can use LLMs, large language models, now to understand what is the patient per site per month to give us a much more idea on how fast the recruitment is going to be. Next. As I have said earlier, anomaly detection is a very big part of a statistician's work. It's no longer that before, we just wait until we see all the data and then we'll start cleaning the data and see if it works. But the truth is that there is anomaly it may not necessarily be with the intent of fraud, but there is anomaly in data every single time. So for example, it could be that the patient is entering the measurement, or the clinician is entering the measurement wrongly in a particular device, or a patient is non-compliant with a particular device. For example, if there is a diary that we give the patient and then the patient has to enter a particular measurement in this diary and that patient is not complying with that diary, that means that there's going to be a lot of missing data and so on. It could be as well that the measurements are not going in the same direction. Take, for example, let's say one measurement should be correlated with another measurement, and all of a sudden, these two things are actually not working very well. So potentially, One is it's wrong. The sites were trained wrongly. The clinicians are trained wrongly. Or it could be that, they're just not doing their jobs very well. So all of these things have to be looked at every single time in such a way that we are able to measure the effect of the drug with precision, with accuracy, and therefore the data is trustworthy to be able to support a particular indication. Just imagine, while a particular drug is not like rocket science, it's not big, it's a very small pill, it's an injection and so on. It doesn't look like Elon Musk's SpaceX, going into the space. But there is a lot of science that is involved in that single pill. And secondly, you wouldn't want that pill to go into your body knowing that it does not have adequate, robust, and reliable data. So we wanted to be able to make sure that the data backing up the efficacy and safety of that product is actually reliable. So, just wanted to put this into context that while a particular pill is not large, it's not, it's not explosive. It is nonetheless, there's lots of science involved in it. And we have to make sure that the data that is used in that particular science is actually reliable. And then lastly in here is the analysis standards, because for me, this is very important, and one where so there are three things that could potentially happen in here. A statistician generally writes a statistical analysis plan. That analysis plan is transferred into a programming language for estimation. And then subsequently, there is what we call reporting, which is whatever the results of that analysis is reported into a particular table. Any of these three processes could actually go wrong. Potentially, it could be that what was written in the statistical analysis plan was written wrongly, and therefore it is interpreted wrongly, and therefore the reporting is also wrong. It could be that it was written correctly, but then the estimation, the programming, actually there is something wrong with it. Maybe not with intent, but of course there's these programs are actually huge with tens of thousands of lines. So you could just imagine that any of these things could go wrong at one particular instant. And the last one could be that if the estimation is very correct, the program is correct, then the wrong number was actually lifted from that analysis into the table. And so then that also becomes a problem, no? Because then we would be reporting something that is not consistent with what was written, nor not consistent with what was the intent of the program. So a lot of these things have to be standardized in such a way that we will minimize the errors that we have whenever we are submitting these analysis or we are writing these reports into a particular manuscript or so. In many of these things, artificial intelligence, including retrieval augmented generation, is already being tapped to help automate many of these things, as well as understanding whether we can improve quality of the writing of the SAP, the analysis, and as well as the reporting of the results into a particular table. So those are just some of the things that a statistician or a quantitative scientist reporting to the role of a statistician is doing. So it's becoming increasingly complex. However, I always think of it as with the age of AI, we are now able to unlock a lot of insights out of data, as well as in multiple types of data sets, including biomarkers, real-world data, and so on. And so I always tell everyone that in the current age where AI is dominating many of the things that we are doing, what happens is that data is integrating. So the role of a statistician has to integrate data, meaning multiple types of data. The techniques that we have to do as a statistician or data scientist also has changed into an integrative one. And at the same time, we have to increasingly use data visualization as a powerful way of making results of the analysis much more consumable to stakeholders. Because at the end of the day, it is the clinician, or is it the physician, or it is the regulator that interprets the data that we have. Much as I would want a statistician to interpret the data as well. But of course, from a translation perspective, these cross-functional roles have to play in part too. Next, please. So I'm going to provide some of the very detailed examples of statistical problems that we do in clinical development. Some of these I have already talked about in the previous slides, but I'm going to put a lot more color and detail into it. So for example, in recruitment, randomization, diversity, and optimal allocation, it could be, let's say, in multiplicity. Multiplicity is making sure that we're not making dubious claims into the data that we see or the results that we see, as well as in pharmacovigilance, let's say post-marketing trials. Next, please. So one of the very, as I've said earlier, one of the very important things that a statistician or quantitative scientist working as a statistician has to do is on patient recruitment. So there are 4 stages in patient recruitment. One is the arrival at the site. The next one is the screening process, meaning whether the patient has the disease that is required, and then remain at trial, which is at that time we are monitoring things, and then completion of the patient, which is last subject, last visit. So in a particular trial duration, it always starts with site activation, and then we have first patient enrolled, and then we have last patient, and then we have last patient visit. What is important to us in general is when the first patient is enrolled and when the last patient is enrolled. And the reason why is that we can predict already all the other things, right? For example, when would be the last patient visit? If we know when was the last patient that was enrolled. So in any enrollment model, usually what we are concerned about is in stage 1 and stage 2. However, there are certain cases as well. For example, if we are interested in understanding whether we have sufficient number of patients with certain duration, let's say 1 year duration, because that would inform us on the safety of a particular product in the long term. So that's a very different question, which I'm not going to be talking about today. So in these recruitment models, I'm just going to be looking into stage 1 and stage 2. Next, please. So there are lots of patient enrollment models out there. All of these are listed in this table here, but I'm only going to focus on 2 things. One is the method by Anisimov and Fedorov in 2007, and then Zhang and Long in 2010. In these models, for Anisimov and Fedorov, they used a constant rate model, meaning that the recruitment rate is constant. It doesn't change. Whereas in the next model by Zhang and Long, it is time varying. Things change. In general, there is some merit to a constant recruitment rate if, for example, all the sites are activated at the same time, right? But problem is that not many sites of a particular trial are activated at the same time. So a trial is composed of many sites. So we recruit, let's say we recruit from Brazil, we recruit from the Philippines, we recruit from China, we recruit from Japan, the USA, or anywhere in Europe. The problem is that most of these sites don't start at the same time. The reason for that is the IRBs for each of the sites may not necessarily have the same schedule. So for example, in the United States, the IRB is a lot faster than when I am going to recruit a trial in Brazil, which takes about a year to get IRB approval. So most of the trials do not have the same number of sites in any particular time. And therefore, the constant assumption may also be a little bit a very strong assumption, whereas a time-varying assumption on the rate of the enrollment may probably be better. But then I'm going to tell you on that one later that it's also not the truth. It may not necessarily be the truth either. So next, please. So this is just a particular case study, a phase 2B multi-center dose-ranging trial to evaluate the efficacy and safety of a particular product. We call it Treatment X in participants with active systemic lupus erythematosus, or SLE. I think this, for all of you who are aware, this is probably what Chris Aquino is suffering. She has lupus. So you probably know that this is really a very serious disease. And therefore, one of the things that have a lot of unmet medical need, and therefore we are constantly trying to create medicines that would help these patients. Another thing is that these patients, this particular disease is very difficult to treat because it's very heterogeneous, as I'm going to show you later. So in this particular phase 2 trial, again, this is still phase 2, this is not yet phase 3. We have 23 countries, total number of countries. We have 107 sites with at least one patient. So you can already see that this is a global endeavor. And it's really very complex, of course, because there are multiple sites. And the total number of patients randomized is 350. That is what we wanted. Here, in this particular case, we are not considering the screening of process, which potentially could be a lot bigger. So, for example, we would be screening, say, maybe 500 patients. That's just to be able to get 350, just because most of these patients will potentially violate one of the inclusion or exclusion criteria. And you can see in here the cumulative number of enrollment on the right after the first site was actually open. And as you can see, because the sites don't open at the same time, most of the time it's usually the enrollment picks up at a particular time point, which is usually a little bit later. And not that we will be recruiting things, recruiting participants constantly. Next, please. So this is just an estimation of the recruitment process. The x-axis is in time, which is in months, and then the y-axis is the enrolled number of patients cumulative. We are interested in 350 patients. What these things are actually saying is that we will be able to complete the trial in around 40 months, as what this graph is using. But then, what we are interested in is prediction, not necessarily, analysis. For us, analysis, after the trial has already completed, it doesn't make a lot of sense, right? What we wanted to be able to do is we wanted to understand when the trial is going to complete. So, for example, if I am at the middle of the trial, let's say, can you go to the next slide? If I am at the middle of the trial, let's say I have already accumulated 200 patients. When would be the time that I will accumulate the remaining 150 patients? I wanted to be able to predict that because I wanted to understand when my trial is going to be completing. So in here, the actual last subject first visit, meaning that the last subject enrolled, it took 15.8 months to do that. And then the actual enrollment period, meaning for this particular trial, took 41 months. Now, the question is, can we predict that it is 41 months using these models? Next, please. Can you go next? So as I have said, there are two models. Can you go next? There are two models that we use. One is the constant rate, which is the homogeneous Poisson process, and the non-constant rate, which is the non-homogeneous Poisson process model. And as you can see in here, the predicted time to enroll 300 patients when we have already completed 200 patients is 40.5 for the homogeneous Poisson. And the 50% confidence interval is 39 and 41. On the other hand, for the non-homogeneous Poisson, we are seeing, can you go next? I think this is a slide, Bill. We say that it's 42 months with a confidence, 50% confidence interval of 41.1 and 42.8. One of the notes in here is that although the homogeneous Poisson process appears to have poor fitting, its prediction is actually not very bad at all. Yeah. On the other hand, the non-homogeneous Poisson has good fit, but it's also bad as well when it comes to prediction because it's only fitting the training data and not fitting the prediction data, the validation data very well. So nevertheless, I think the model is already informative from my perspective. In drug development, we are not necessarily very interested in things to be very exact. Sometimes some data is already adequate enough for us to be able to make a decision. So when I was asking a particular statistician for this, the first thing that he gave me was a point estimate with a 95% confidence interval. Of course, the 95% confidence interval is going to be very wide. And I told him, nobody's going to believe you if you're going to show this to a physician or a clinical operation because they probably are going to say, I could already predict things and without having to do any math. So we have to be very cognizant of all of these things as well, making sure that we don't look naive when it comes to showing results to physicians as well as in clinical operation. So I told the statistician, I'm fine with if you can tell me that 50% of the time the trial is going to complete within this month or 2 months. That should be enough for me. But I don't think that I would want you to tell me that the trial would finish, with a confidence interval of 6 months, because that's not helping me at all. So these are just some things that we have to be very cognizant as quantitative scientists, because while we are making estimation and precision, quantity uncertainty quantification very well, it may not necessarily be what is needed to answer a particular question. Next, please. All right. So we now go to we call this multiplicity in optimization. So in clinical trials, there are multiple endpoints, right? There are multiple endpoints that we use. So for example, let's say in COPD, which is chronic obstructive pulmonary disease, we would be looking at, let's say, FEV1, which is the forced expiratory volume change. We could be looking at the annualized exacerbation rate or hospitalization in these patients, which are quite fragile. It could be, an endpoint could be a particular PRO that measures the patient, how the patient functions, feels, or survives, and so on. So there are multiple things that we look at. The problem is that the FDA or the EMEA, which is the European Medicines Agency, will not let you do that without multiplicity or control of the family-wise type 1 error. What does type 1 error mean? Type 1 error means that we are concluding a particular drug as effective when in fact it is not effective. And by the fact that we are looking at multiple endpoints, there is a possibility of making a false conclusion in many of those endpoints just because we are looking at. Take, for example, you go with your labs, you go to your doctor. All of a sudden, because there are so many laboratory parameters that are there, it's very easy to have 1 or 2 that actually is above normal or below normal and so on. So that's the same thing. And that could actually be false positive. No, it's not necessarily that all of the time those are actually, the signals are clear and it's indicative of a particular disease. Much like in clinical trials, whenever we're looking at multiple endpoints, there is a potential of concluding that the drug is actually benefiting in this providing benefit in this particular endpoint when in fact it is not. And the FDA is actually very good at making sure that, pharmaceutical companies do not advertise a particular product more than what is really true. So we cannot have a radio station saying that, hey, this is good for diabetes or whatever. As you can probably already hear from many of the things that you hear here in the United States and in Europe, we cannot promote certain things that are not necessarily backed up by robust data. And I hope that in the Philippines, eventually we can do that as well. So we're not going to be confusing many of our patients into spending something that is not really helping them at all with their disease. So, as I've said, there are multiple endpoints. This is one of the products that I worked on before on baricitinib for alopecia areata. Alopecia areata is a condition where the immune system is actually targeting the follicles of your hair, and therefore you're losing hair. Many patients actually have, it's a large patient population, and I'm pretty sure many of you who are listening to this particular webinar have one person or so that having alopecia areata. A related condition on this one would be vitiligo, which is the discoloration, the pig on the skin, only that it's happening on the skin and not on the follicles of the hair. They should have very similar mechanism. And in this particular disease, we looked at multiple things. Let's say SALT is a measure of how expansive the scalp is actually affected. So for example, a SALT 20 would mean that 20% of the hair is affected, or the scalp is affected. Whereas a SALT 80 would mean that almost all of your scalp on your head is affected. And there are other things as well that we look at. Next, please. Now, the question is then, how do we test this particular, how do we make sure that we are controlling the type 1 error of when we are making all of these claims? So I will run first on a few basics that could help us understand how a statistician actually does this. So we call this testing strategy as the alpha saved and alpha lost. So suppose we have two endpoints, A and B, and then we divide the type 1 error. Typically, a type 1 error level is 0.05, two-sided. So we can allocate half of that type 1 error into endpoint A and half of that type 1 error into endpoint B. So meaning 0.025 two-sided. Now, in this particular case, if A is successful, we will transfer all the alpha that was saved into the next endpoint, B. And subsequently, if B is positive, means we're able to reject the null hypothesis, we're able to transfer back to A in such a way that each of the hypotheses is actually tested at level 0.05 two-sided, which is a little bit higher. So this is just the typical, what we call this one is the Holmes test for two hypotheses. Now, if suppose we have three endpoints, A, B, and C, we could potentially sequence these tests as we will test A first at the 0.05 level two-sided. And then if that one is successful, we'll transfer everything to B. And then if that one is successful, we'll transfer everything to C. Right? So this is what we call fixed sequence, because we are sequencing A, B, and C, or testing B only after A is positive, and testing C only if A and B are positive. The problem with this particular test is that it's very inefficient because sometimes we don't necessarily know whether which among the hypotheses is the most powerful. Is it A? Is it B? Is it C? If B, for example, is the most powerful and A is not, and then we put everything, all our eggs in our basket into A, then potentially we could never test B and C anymore subsequently because we have used up all the alpha level on the first endpoint. Next, please. So we have this So we have this what we call fixed sequence and fallback strategy. So we can potentially divide the initial alpha allocation, which is 0.05 into 3 parts. One could be 0.03, one could be 0.01, and one could be 0.01. In such a way that if A is successful, we can transfer everything to B. And therefore, B will be tested at 0.3 and 0.1, which is 0.04. And then if B is successful, we can test C with a full 0.05 level. Next, please. We can further complicate this one by this, what we call extension of the fallback procedure, meaning that if C is successful, I can retest B back with half of the 0.01. So I will test B with 0.01, sorry, I have, that would be 0.01, no, 0.035. So let's go through the logic in here. A and B can be retested at higher levels. So for example, A can be retested at 0.03 plus 1/2 of 0.01 because that is coming from C, which is meaning that it's 0.035. Secondly, if A is successful, B can be tested at 0.035 plus 0.01, which is what B was allocated, and then 1/2 of 0.01, which is then we are testing B at 0.005 level, and so on. Now, what if there are more endpoints? It becomes extremely difficult when we go next. Suppose, for example, we have 4 endpoints, and we have this particular graph. So now the work of a statistician is becoming much more difficult, or any quantitative data scientist. So here we have to be able to put forth what is alpha and what is the transition matrix G. Now, next. We can use, the problem with this particular approach is that each endpoints are not the same. So for example, there are some endpoints that are actually much more valuable than the others. Take, for example, atopic dermatitis. In atopic dermatitis, the more valuable endpoint is what we call the eczema severity index, meaning measurement of how severe your eczema is with the body surface area. Another endpoint would be NRS, which is the pruritus itch scale, meaning how severe is your itching. So that's secondary to the eczema severity score. So in terms of value, I wanted the eczema severity score first before PP-NRS in terms of value. So then it's not necessarily that we have to sequence a particular set of endpoints in such a way that we are sequencing it in terms of power. It could be that I'm sequencing things in terms of value as well. So there are now 2 things that are happening, power as well as a value of the particular endpoint. So we created this objective solution by creating a particular gain function that measures both the power and at the same time the value of the particular endpoint. Next, please. I need to make sure that I think I'll just go straight. What we did in here is that any function can actually be approximated by an artificial neural network, ANN, feed-forward neural network. So I wanted to make sure that that is clear. Any function can be approximated by a neural network. And therefore, once it is a neural network, it becomes a machine learning problem. Next, please. So this is just, what I'm saying, that F, alpha, and G can be approximated by A and N, alpha and G, and therefore, the optimization of the function F is the same as the optimization of the function A and N, right? The artificial neural network function. And therefore, we can gain what would be the alpha as well as the graph, the transition matrix, or the weights that would propagate from one endpoint to another. Next. Next, please. Next. So we tried to do this in a particular example. I'll go back to going back to lupus. Lupus is an autoimmune disease, and it's causing inflammation to various parts of the body, including the skin, the joints, the lungs, the heart, the blood cells, the brain, etc. In this particular trial, we are comparing two dose levels of a particular drug in relation to placebo. The endpoints we're talking about are systemic lupus erythematosus responder index. We call it SRI-4. And then we have the British Isles Lupus Assessment Group-based Composite Lupus Assessment, or BICLA. And we have incidence of flare event, which is just flare. So because there are two doses and there are three endpoints, then therefore there are 6 hypotheses, right? 3 times 2. Next, please. So these are just some of the simulation parameters that we are working on, that we used. Suppose these are the treatment rates and then the rate of the standard of care in each of these hypotheses and each of these endpoints, and what is the power associated with this rates, and also the gain, which is in red. So we say that the gain function for the primary endpoint is 30%, meaning it's the most important. Also, the gain function for the other low dose is 40%, probably because we wanted a much more safer drug. That's why we are valuing the low dose much more. And then for the other endpoints, that would be 0.10, 0.02, 0.15, 0.3. And we have correlations for each of these measures as well. Next, please. This is one candidate design of the graph, which is a little bit complex, no? And it's fairly saturated. You know, you have connections in almost every single thing. Now, is this the most optimal? Next, please. Once we have performed all of these artificial neural networks, so there are 3 possible strategies. One would be we will sequence everything from H11 all the way to H23, meaning that once H11 is positive, we will transfer that to the next endpoint, then we will transfer to the next endpoint, and so on. It could be that we will split 0.05 into 2 from H11 and H21. That would be strategy 2. And it could be strategy 3, which is the optimal strategy that was determined by the artificial neural network. Next, please. So what would that mean in terms of being able to accept or reject a particular hypothesis, which is related to the endpoint? In strategy 1, we are only able to reject 2 hypotheses, the first one and the second one, H11 and H12. The strategy 2, we were able to reject 2 hypotheses as well. But in the optimal strategy, we were able to reject almost all except for H13. So in this particular case, the expected gain is actually higher in the optimal strategy as opposed to strategy 1 and strategy 2, meaning that there is a possibility of us being able to negotiate with the FDA or EMA on many of these endpoints in the label. Again, in the United States, we cannot market a particular product with respect to a particular claim if that claim is not substantiated. And particularly if that claim is actually not adjusted for multiplicity. In this particular case, that's what we are doing in here. So in the optimal strategy, we have a chance to negotiate with the FDA of being able to market the claims of that particular product. So for example, in this particular thing, we will be able to market that, hey, it's able to reduce the incidence of flare, or let's say it is able to reduce the activity of the disease based on the BICLA score and so on. So these kinds of things. Now, I know that, I'm almost there, but I wanted to make sure that we have time for questions. So I'm just going to stop in here. Again, these are just some things that a statistician or a quantitative scientist working as a statistician does. As I've said previously, it's not that, the statistician has to be a statistician by training. It could be, for example, I have a statistician whose training was a veterinarian a long time ago. I have a statistician who is actually a molecular biology, has a PhD in molecular engineering, biotechnology engineering. So just because he or she is very quantitative, then he is able to perform as the statistician. So it's not necessarily that the training has to be matching the role. It could be that you're a mathematician. In fact, I have one mathematician currently working on anomaly detection. So he's also working as a statistician, and particularly on anomaly detection, because he knows variational autoencoders, as well as all of these diffusion models that he used when he was writing his PhD on flow cytometry and so on. So with that, I'm going to stop here so that we have enough time for questions. And thank you.
Thank you, Meg. We're now accepting questions from our Zoom audience. Anybody? So just unmute your microphone, please, and ask your question.
Don't be
So, hi. So, Meg, Dr. Gamalo, you mentioned before yung AI. To what extent are you using, or is the use of AI technology in particular in your industry? So
Yeah, that's a very good question, actually. That's really a very good question. And in fact, that's one of the things that I'll be talking about in 2 weeks and in next month in another conference. There are many ways for which AI is actually being used in drug development, but we can think of it in terms of this way. Where is the most valuable use of AI in drug development? Currently, the most valuable use of AI in drug development is in drug discovery. So meaning the creation of the molecule itself, whether that particular molecule binds into the target, and then whether the target actually is eliciting a particular response consistent with, modulating a particular disease, and so on. So that is one that is very active currently, and it's the one that is most valuable. And why do I say that? It takes a very long time to create a molecule. Secondly, most molecules are actually duds. They don't necessarily go into a clinic because, for example, it could be that the toxicity is actually not favorable for the compound to go into the clinic. So that is one that is really very, I would say, very advanced. The other one is on document simplification and automation. So we do, as we know, large language models is very powerful. ChatGPT and so on, including DeepSeek and so on, they are very powerful, and they are able to streamline many of the things that we are doing. Now, there are other places where we are using drug development or AI. As you have seen, we can do large language models with network or target product profile, disease progressions, extraction of information from, let's say, public sources, scraping, and so on. Those are actually very valuable, but the impact is small. The impact is small. And the reason why the impact is small is that drug development is a highly regulated process, meaning that we have to go through a sequence of steps to be able to have the drug approved. And in each sequence of steps, there are rigorous processes involved. So even, for example, in programming, we can probably, let's say, automate certain programming things, right? So for example, I could probably ask ChatGPT, hey, write a code for me to be able to do this analysis, blah, 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 blah. The problem is that analysis actually the correct thing that was stated in the SAP? Is that analysis actually QC validated? Is the reporting of the results of that analysis QC'd and validated? So it requires more levels of things. Now you can probably say, hey, I can use multiple agents in that one too, right? I could use several agents to do all of these things. But we're not yet there. What I mean is that we're not yet there where we're very comfortable that these things are actually producing something that is correct. And it would be a nightmare if we send all of these analyses and results to the FDA or EFEN, and all of a sudden the FDA says, oh, this is wrong. Because then just imagine that they would be saying that, return to sender. We cannot believe these results, and therefore it delays the approval of a particular product. And just imagine as well, for the public who is actually taking this drug, the public wants to be sure that the data, the analysis, and the interpretation of that analysis is actually accurate. Just because I am ingesting that particular product, which affects my health and which affects my safety. So it requires a lot of things. So I'm not saying that there's not a lot of applications of AI. There are plenty, and I could see it everywhere. And in fact, in my team, the way that I've been presenting this is that a traditional statistician may have to embrace methods that are non-traditional, includes machine learning, artificial intelligence. These are non-traditional things, including large language models, to be able to do their jobs currently. So it's no longer sufficient, I'm pretty sure, in the near future, to be just trained in the classical statistical training. That answers the question.
Yeah. So any other question? I'm just wondering, Meg. So maybe early in your career, or even now,
Yeah.
to what extent do you need to be familiar with all sorts of sorry, I just have to adjust something here.
No worries. You mean familiar with drug development processes?
All familiarity say biotechnology or molecular biology, so that you'll be able to ask the correct questions.
So that's a very good question. Yes, sometimes it is necessary, but I wouldn't dismiss anybody just because he or she does not have those kinds of training. And the reason is it really depends on what a particular job requires. Most of the time in drug development, even for me, I was not trained as as somebody who would be working on drug development. My discipline was in statistics, particularly in functional magnetic resonance imaging. I never had anything on clinical trials when I started my career 20 years ago. If I remember correctly, my boss, when he hired me, he gave me a few documents the very first day that I entered my job. And those documents were International Conference on Harmonization, ICH E6, E9, E11, E10. So I would say that nobody really is trained exactly as how the job needs. I think what is very important is a colleague that has agile, that is able to change things very quickly in order to perform the job, and then learn from that job very quickly as well. I think that's the most important, because nobody will be trained exactly as how the job would really need.
Questions? So, okay, yeah, Ray. No questions in. Yeah, Carlo.
No, boss, sorry, pahabol. Mike, I'm curious about data handling, specifically, how do you detect errors in data, or how do you flag data that you receive maybe from participants in clinical trials as fraudulent? And is it more of an expertise sort of thing, or do you use automated tech?
That's a very good question. In fact, that was asked of me when I was in the conference in Society of Clinical Trials. I would say two ways. So one is domain expertise. So for example, we would be looking at data that, well, they're supposed to be going together, and then all of a sudden, we're seeing things that are a little bit not that way. So we would use kind of like supervised learning, unsupervised learning as well, to be able to understand clusters of data behaviors and so on. It could be as well that again, it could be supervised and unsupervised. The unsupervised ones, we will cluster the data and then see whether there are certain clusters that are unusual. And also, not only that they are unusual, to be honest, some of the anomalous things are when the variability is actually very smooth, meaning that there seems to be nothing going on. So the data is just very clean. That actually tells you as well that, oh, there must be something that is fraudulent on this one, because the human behavior is that we are only limited in the things that we actually can manipulate. So most of the time, for example, people with fraudulent intent only use certain numbers. We don't use the whole set of 10 numbers at all in digits and so on. So there's only a few things. So whenever variability is also low, that's another problem. Now, can we automate certain things? For example, if I don't know any domain knowledge in that particular disease, can I automate it? And the answer is not very sure. No, I mean, we can potentially do that with large language models, because large language models already have some reasoning in it. For example, hey, among all of these things, what should I be doing? And then after that, create certain processes for certain things that was actually enumerated in that response and so on. So maybe it is. Maybe it can be done. Is it there yet? Maybe not yet. Again, this was a question that was thrown to me previously, and I was like, okay, yeah, it made me think a little bit, but yes, potentially large language models can help to identify which are the ones that I truly am going to be looking at, and then monitor. Now, the question is, the problem is that large language models generally give you only what we call the expected ones, right? Expected ones, because it usually tells you, the more common latent spaces. The question is whether can it actually give you something that's unexpected as well. And I'm not sure. I'm not sure yet on that particular question. If, let's say, maybe you can prompt what are the unexpected potential anomalies they have to take a look at, and then from then build on that particular response. I'm pretty sure we will be there eventually, but I haven't seen it yet.
Right. Thank you.
Mhm.
Okay. Ray?
Okay, thanks everyone. Before we let you go, we just wanna promote, because we have, just share this first. Yeah, before we end, we just wanna promote a new IE that we're offering this first semester. So if you're interested in about applying statistics in pharmaceutical clinical trials, please check out Math 60.3i. So it's being developed by Pfizer together with the math department. So, and it will be every Wednesday mornings if you're interested in actually taking it up. So thanks everyone. Thank you, Meg. Thank you, Sir Russell, for having us for
Thank
sharing.
you very much for having me as well. I would really encourage everyone as well on this particular course. Again, when I was starting my career, I never had this, and I wish I do. Because then it would have given me a much more, some readiness when I enter the workspace. So
thank
that's all.
you.
Thank you.
We
Thank you,
hope to
ma'am.
see
Thank
you
you.
during the first semester. Thanks, everyone.
Okay, bye-bye.
Bye.
Ayan,
Thanks,
pagod na.
Joe. Thanks, everyone.